Hi, this is Dr RAFIQ Sir, MSc PhD Life Sciences. I was the recipient of several national recognized fellowships provided by the UGC, and I published almost 15 research papers, several books, and I am involved in this profession, in this teaching profession from last 10 12 years. And I'm also the recipient of several best teacher awards. So I'm preparing the students for their competitive exams like NEET, like JEE, like PG entrances, or any other professional exams. So I'm involved in this preparation for last 10 years, and I feel very much energetic in this profession. And with the Baiju, this is my first experience, and I am taking you directly to the demo lecture, and which I chose. Hope the board is clear. Which I chose. That's the DNA as the genetic material. What's the full form of DNA? This is deoxyribonucleic acid as genetic material. I choose this topic because this is a very basic and simple topic, and in this topic, I would take you to the several experiments that led to the discovery of the DNA as a genetic material. So let's talk about the DNA first. So DNA, its full form is deoxyribonucleic acid. Deoxyribonucleic acid, and it's called the master molecule of life. It is the master molecule of life. Why it's master molecule of life? Because it is involved. It regulates all the functions of the cell. It is the DNA that decides that which thing is required by the cell and which thing is not required by the cell. So coming to the genetic material, so genetic material is any substance that has ability to move from one generation to another generation and to carry the inherent characters or to transmit the inherent characters from one generation to another generation. So it's a very brief lecture, as I have been told that I have to record the lecture for just 5 to 7 minutes. So I would directly take you to the experiments. So first experiments in this direction that proves that DNA is a genetic material, that was basically performed by the Frederick Griffith, and it's called Frederick Griffith transformation experiment. He did this experiment in 1928, and he chose the bacterium, that's Diplococcus pneumoniae or Streptococcus pneumoniae. So this is the bacterium that was chosen by the Frederick Griffith for his experimental purposes to prove that DNA is a genetic material. See, so any organism that is chosen by the scientists for their experimental purposes, as I do in my research tenure, we call it as model organism. We call it a model organism. So here, Diplococcus pneumoniae or Streptococcus pneumoniae is the model organism that was chosen by the Frederick Griffith in the transformation experiment to just prove that DNA as a genetic material. So basically, let's know something basic about this bacterium. Here, it is Diplococcus. It is di means two, and coccus means a spherical shape. So the shape will be like this, and this bacterium is the causative agent to cause a disease called pneumonia. What's pneumonia? That is just the respiratory disorder, as the coronavirus did in the organisms right now in the world. That is basically the respiratory disease. So this bacterium is basically having two serotypes. One serotype is R second serotype and another is S third serotype. R second serotype and S third, we call them serotypes. Now what are serotypes? Serotypes are basically immunologically differentiated species. See, it was basically sometimes before known that the serotypes Serotypes are basically immunologically differentiated species. Now, what is the meaning of this term? Serotype. See, these two serotypes, S R second and S third, our immune system respond them differently, because this R second, this is the virulent strain. Virulent strain means this strain cannot cause the pneumonia in the organisms. However, the S third strain is a virulent strain because it can cause the disease in the organisms. So how you can differentiate between R second and S third? See, I am talking about the Federer experiment, transplant experiment. But before going into the experiment, I am telling you some basics about this model organism that was chosen by the Federer group, and that's a Diplococcus pneumoniae or Streptococcus pneumoniae. I am saying that this type of organism is having two types of serotypes: R second serotype, S third serotype. Now, what are serotypes? Immunologically differentiated species, means our immune system responds these two strains differently. And what is that response? R second strain, it cannot cause the disease. And what is that disease? That's pneumonia. But S third can cause disease, and what is that disease? That's pneumonia. So how you can differentiate these two species? See, I'm showing you experimentally because this is very important. See, if we prepare an artificial growth media, artificial growth medium. What's artificial growth medium? That is when you are preparing some growth medium artificially inside the lab, or we should say it is simply the in vitro culture. In vitro culture of the Diplococcus pneumoniae bacterium. Means you are adding all the required nutrients into the starter plate. Means this plate contains both micro as well as micronutrients. You are making a solution of these micronutrients in a simple glass plate, and after that you are adding the distilled water plus you are adding the necessary hormones into this plate, and finally you are adding the agar solution. What's agar? Agar is basically the solidifying agent. That's basically the powder that's extracted from some algae from their cell walls, especially the red algae like chondrus, like Gracilaria, etc. So we are adding agar to this medium just to solidify the medium, because this medium was initially in the liquid form, but we need this medium in the solid form. That's why we are adding the agar powder. So when we adding the agar powder, so it will become gel, and after that we are inoculating this very much bacterium into this plate. And when we adding this Diplococcus pneumoniae bacterium into this plate like this, and this process of adding the Diplococcus pneumoniae is called inoculation. See, here this bacterium is getting all the nutrients readily available, because it is basically the solution of micro and macronutrients. And when this bacterium is getting all the nutrients in the ready-made form, it divides and redivides to increase its population. When it increases population, it forms the colonies, and we have two types of colonies like this. We get two types of bacterial colonies like this. So there is difference in the shape of these colonies. In the one colonies, we are having the rough outline, because the outline is rough. And while in these colonies, the outline is smooth. So these colonies, we can call these colonies as our second serotypes, and with these smooth outline colonies we call the as third serotypes. So this is the method, this is the experimental method, how you can differentiate between the R2 and S3 serotypes. That is, in the R2 strain, you are getting the rough colonies, means the colony that is having the rough outline, and in the smooth colonies, you are getting the smooth outline. And basically, why these are smooth and why these are rough? It is basically the presence and absence of the capsule. Capsule. Now, what's capsule? Capsule is the basically a polysaccharide covering on the cell wall of the bacteria. See, if this is the bacterial cell, and above the cell wall there is a silimy covering. Silimy covering is basically this silimy covering called a capsule. It's basically formed of the polysaccharide. And what is the function of this capsule? Its function is simply to protect the bacteria from the attack of antibiotics. So in certain bacteria, this capsule is present. In certain bacteria, the capsule is absent. And in certain bacteria where capsule is present, the outline is obviously smooth and weak, and that is the S strain. While in certain bacteria, this capsule is absent and the outline becomes rough and weak, all that the R strain. So this is basically the beginning of this experiment. And what Griffith did, actually, this is my demo lecture. I don't go on to details. It will become the more lengthy lecture then. So I think this was my initially the demo lecture. Hope you people understand what my depth of knowledge is, and hopefully you people will consider me for this, for the academic specialist. I hope so. Thank you.